Hi everyone, welcome to the first week of Unit 7 where we will be exploring the major body systems. We won't cover everything, but we're going to cover the major highlights, starting out with respiratory and circulatory systems. Now, respiration and circulation work together to accomplish gas exchange. All animals are aerobic, meaning that they require oxygen to produce energy, and the energy that we produce is called ATP. If you remember from last semester, ATP stands for adenosine triphosphate, meaning it has 1, 2, 3 phosphate groups, and that last bond is readily broken to release a lot of energy. In fact, a little bit more energy than it costs to make the bond. So we need oxygen for this process. This is one of the major challenges facing all multicellular animals, is getting enough oxygen to make enough energy, ATP, and then getting rid of carbon dioxide. So this process is going to happen in two directions. In the lungs or the gills, there's going to be gas exchange where oxygen enters into the blood and carbon dioxide leaves the blood to be exhaled. But in the body tissues, gas exchange happens in the opposite direction, where oxygen leaves the blood to go into the tissues and carbon dioxide goes into the blood to be exhaled. So why does this happen? Well, we have high levels of oxygen in the lungs or in the water that the gills are in, and there's relatively less oxygen within the blood, which means that we've already have a concentration gradient. And these gases, they only move by simple or passive diffusion. So they have to move with the concentration gradient. Remember, we're going to go from an area of high concentration to an area of low concentration. So that's why this happens opposite in the body tissues as the lungs or the gills. Because in the lungs and gills, we have high oxygen pushing its way into the blood. Whereas in the body tissues, there's low amounts of oxygen, so the oxygen is pulled out of the blood. This is an example of an alveoli. Now, we will talk about gills, but I wanted to talk about the alveoli in a second. Lungs, like human lungs, are made up of a bunch of tiny little sacs called alveoli, and these sacs are inflated. Okay, there's a thin layer of surfactant fluid along the membrane of the alveoli, which serves two purposes. One, it creates a little bit of surface tension to allow the alveoli to stay inflated. The other is that it allows these gases to dissolve. Dissolving the gases in this fluid allows it to more easily transfuse into and out of the capillary beds. Speaking of capillary beds, this is what those alveoli look like. Here, this is a bronchiole, which will go into the bronchi, which will go into the trachea to be breathed out through the mouth or nose. But what I wanted to bring your attention to is these capillaries. Capillaries are very, very, very small blood vessels. In fact, an erythrocyte or a red blood cell can only fit single file through a capillary. And notice that these alveoli are completely surrounded by capillaries. This allows for lots of gas exchange. So we have high concentrations of oxygen in the alveoli from breathing in, which means that oxygen will be entering into these capillaries to be distributed throughout the body. But there's high levels of CO2 in this blood. So as this blood enters these capillaries, CO2 is exiting to be exhaled. All right, so what exactly is the air pressure? So we mentioned that the oxygen has to kind of push its way into your tissues. Well, it does that because we have air pressure. At sea level, if I'm sitting here at sea level, the atmospheric pressure is about 760 millimeters of mercury. Now, why would we measure it in millimeters of mercury? That seems like a weird way to measure pressure instead of like PSI or something like that. Well, it's from this classic experiment. So if I have a vial and the vial is filled with a thick, dense fluid such as mercury, the atmosphere is going to be pushing down on that fluid that's open. So here's the vial, it's upside down in kind of a petri dish, and the atmosphere is pushing down on it with a force. And that force is enough force to push the liquid up the vial, up this column, till about 760 millimeters. So this is 76 centimeters or 760 millimeters. This is your air pressure at sea level. But if I climb up this mountain and I go up to the top of Mount Everest, now I have more or less air pressure. Well, the air pressure is a measurement of all of the particles that are on top of your head. So if I'm standing at sea level, you see all these particles. There's tons and tons of particles exerting force downwards due to gravity. But if I climb all the way up this mountain, I no longer have all of these particles, and now I'm just focused on these particles. So there's overall less particles pushing down on my head. So there's going to be less air pressure. In this case, there's about 401 millimeters of mercury. Now, when I'm at the top of Mount Everest, I'm going to need a supplemental oxygen mask. Okay, it's not a question. I'm going to need it. But why am I going to need it? Well, there's about 21% of the atmosphere that's made up of oxygen, which is great. That means that if I multiply 21% by 760 milliliters of mercury, my partial pressure for oxygen is almost 160 millimeters of mercury. That's a lot of pressure pushing the oxygen into your lungs. Now, most of the atmosphere is made up of nitrogen. This is a very neutral gas. About 80% of the air that we breathe has nitrogen in it, but it's non-reactive. We don't use it for gas exchange. A very, very, very small proportion of the atmosphere is made up of carbon dioxide, which is really great for us because we're exhaling carbon dioxide. So it's going to be much easier for us since there's not that much carbon dioxide there. Now, if I'm trying to recalculate my partial pressure, I need to multiply the new atmospheric pressure at the top of Mount Everest, which is 401, times the percent of the atmosphere that is oxygen, which is 21. So whereas at sea level, I have 160 millimeters of mercury pressure pushing into my lungs, now I only have 84, which is significantly less. So I'm going to need a supplemental oxygen mask, not because there's less proportion of oxygen, but because there's less atmosphere altogether to be pushing that oxygen into your lungs. All right, so if we were able to manipulate this, how could we increase the rate? Because that doesn't seem like a very efficient process. So that leads us to Fick's law of diffusion. Fick's law of diffusion estimates the rate that diffusion happens. How quickly does it happen? And the rate is dependent on the diffusion constant. So we can't really change the diffusion constant. That's going to be the same due to the laws of physics. We can't change that. But there are some things that we can change, like surface area. If we increase the surface area as much as possible, it will increase the rate of diffusion. So anything on the top, we want higher surface area. And if you think about fish gills or even human lungs, you realize that there's lots of effort, evolutionary effort put towards increasing the surface area of these tissues. Next is the partial pressure. Now, we can't really change the atmospheric pressure in the atmosphere, but there are a couple ways that we can change the partial pressure. So we'll talk about that in a little bit. The last one, I'm not quite sure if we can change that. Sometimes we can, sometimes we can't. The last one is distance. We actually want to lower the distance. Usually we're only talking about 1 or 2 cells thick that the oxygen can diffuse through. Any more than that, as we get more and more cells stacked up, it's going to be harder and harder for the oxygen to push its way through or to diffuse through with simple diffusion. So the thinner the membrane, the better. So if we look here, there are 2 major areas that we can do to increase the rate, which is the area and the distance. We want as little distance as possible and as much area as possible. Now, just as a little side note, we're going to come back to this slide later on when we talk about the circulatory system and we talk about blood, but just for a minute, I wanted to talk about erythrocytes or red blood cells. Red blood cells don't have very much going on for them. There's no nucleus. They don't have a lot of the stuff that other cells have, so that they can be very small and fit through those capillaries. That means that they only live for about 120 days and then they need to be replaced. But what I wanted to talk about is hemoglobin. We've been talking about gas exchange. Hemoglobin is a protein found inside of the red blood cells, inside the erythrocytes. In fact, there's over 200 million hemoglobin per red blood cell, which means tons and tons and tons of heme. Now, what is a heme group? Well, hemoglobin is made out of 4 different protein chains, 2 alpha chains and 2 beta chains, and each one of those chains has a heme. In the case of hemoglobin, this heme is made with iron. And remember, iron, when it mixes with oxygen, turns a red color. So this is what gives our blood that red color, but it's also what allows oxygen to be carried through the blood. So oxygen is attracted to the iron in the hemoglobin. All right, but that's not the same with carbon dioxide. Carbon dioxide doesn't necessarily attach directly to the hemoglobin. Sometimes it dissolves in the plasma. Most of the time, it dissolves directly into the blood itself. Only about 20% is attached to the hemoglobin. 8% is dissolved in the plasma. This one says 10%, so 8 to 10%. But the majority of it dissolves directly into the red blood cell, becoming bicarbonate. But the thing that I wanted to talk about is the little bit that's dissolved in the plasma. When carbon dioxide is dissolved in water, it creates carbonic acid. This is going to slightly lower the blood pH, especially in your tissue areas that have more carbon dioxide. So this is going to slightly lower the pH or make it more acidic because of this carbonic acid. So your brain is actually quite sensitive to this acidity. And this is that reflex that makes you feel like you need to gasp for air when maybe you're trying to hold your breath underwater. It's a very uncomfortable feeling, and it's a very strong impulse. Your brain wants you to breathe. So if it's sensing a whole lot of acid in your blood, it's going to start to you're going to start to panic. These signals can tell you to either breathe more or to slow down your breathing, depending on whether you're hypoventilating, which means you're breathing too little, or hyperventilating, meaning you're breathing too much. In both of these cases, you are probably going to need to adjust your breathing rate in order to calm down or to restore your normal CO2 levels in your blood. This is actually really interesting because of something called a Bohr shift. We already talked about how oxygen is attracted to the heme in your blood cells, so the hemoglobin in your blood cells. But this is highly affected by pH and temperature. When there is a lower temperature or a more basic pH, the oxygen affinity is pretty stable. But if we have a warmer temperature or a lower pH or is more acidic, it actually causes the oxygen to release from the hemoglobin, or it actually reduces the affinity for oxygen. So that means in our lungs, where there's not as much carbon dioxide, we have a higher pH or a more basic pH. And this is also a lower temperature just because it's exposed to the air. This means that we're going to have a high affinity for O2, and those blood cells are going to grab onto that oxygen. Now, when it comes to the body tissues, there's a buildup of CO2, meaning a slightly lower pH or slightly more acidic. Also, there's a slightly higher body temperature within the core of the body as opposed to the outside of the body, which means this actually lowers the affinity for oxygen, allowing the oxygen to be released from the blood into the tissues. So really interesting effect that the Bohr shift has. The Bohr shift is going to explain the percent oxygen saturation in the blood as opposed to the partial pressure of the air, of the oxygen. Remember, we want to have about 98 to 100% oxygenation level. So really, we have to have almost like 90 to 100 milliliters of mercury in this zone, okay? Anything less than that, you're going to have a hard time saturating your blood with oxygen. So that's why that Mount Everest, where we had like an 80% saturation level or a partial pressure level of oxygen, it led to less saturation in the blood. But in order to get up here, we want to shift, and the bore shift allows us to do that. When we have lower temperatures or higher pH, we shift this direction, and when we have lower pH and higher temperatures, we shift this way, which means that we're grabbing onto the oxygen and then letting go of the oxygen over here. Now, we already talked about oxygen's affinity to blood and CO2's affinity to blood, but there's another gas molecule that really, really loves hemoglobin, and that is carbon monoxide. Now, carbon monoxide does nothing for you metabolically. You can't use it to make ATP, but the hemoglobin loves carbon monoxide. In fact, once you breathe carbon monoxide, and CO2 is joined to that hemoglobin, it is no longer useful. It will never let go of that CO2. So you would slowly suffocate as more and more blood is occupied by carbon monoxide instead of oxygen. So those blood cells are useless until you replace them and make more blood cells. So sometimes these symptoms can be like a mild headache, short of breath, but honestly, there's no odor. You can't see it. It's colorless, odorless. It can cause dizziness and nausea, but other than that, not easily detected. So this is why it's very important to have regular maintenance, especially on older vehicles, and proper ventilation in enclosed spaces when you're using like a generator or something. But what do you think is the oldest method of detecting carbon monoxide? Well, if you've ever heard the saying canary in a coal mine, the canary in a coal mine is real. We used to use canaries in these enclosed spaces, and when the canaries stopped singing, that signaled to the workers that they should evacuate because there's not enough oxygen available or there's too much carbon monoxide. All right, gas exchange. I want you to be thinking about what you think is the most efficient type of gas exchange. There are many evolutionary changes that have occurred and accomplished this goal of gas exchange in many different ways. Here we can see cutaneous respiration, where we respire directly through the skin into the body cavity, but these organisms are going to be very limited in size. If we want to get larger, we'll have to find a way to get the oxygen inside the body, i.e. the trachea system. This is seen in insects where we just have these tubes, and that's essentially it. They're just open tubes. There's no breathing apparatus except for maybe the insect's movement, normal movement that can cause it to kind of move in and out the oxygen. But other than that, this is just straight diffusion. It's just an advancement of this cutaneous respiration. Larger animals still, like fish, have something called gills, and gills, the capillaries run through these areas with lots and lots and lots of surface area. So you can see here an effort to increase the surface area. But we also have lungs, which have all of these tiny little alveoli or tiny little air sacs, which also increases the surface area, and you can see our blood vessels going through there. So gases can diffuse directly into the organism if they're small enough, but once they start getting bigger, they're going to require more complicated adaptations to achieve that gas exchange. We just went through a few, such as amphibians and planarians using their skin, insects having a tracheal system, fish using gills, and mammals having alveoli. But there is also a way that animals can adjust the partial pressure of the oxygen. So here I have something called co-current flow. For instance, if I'm talking about these gills, I have deoxygenated blood moving this direction across the gills. We see we go from blue to red. But if I'm talking about co-current, that means the water is also moving this direction. So let's just stick with that example. The water and the blood in the gill are going the same direction. Now, if I'm blood in the gill, I'm going to start out with 0 oxygen level, and the new water coming in starts at a 10. We'll just use that as 100%. Now, the pressure gradient, or the partial pressure here, is pushing this direction down into the blood. So we're going to lose an oxygen in the water, and we're going to gain an oxygen in the blood. And I'm going to keep doing that until we reach equilibrium. So it turns out that once I get to about 50%, then we've reached equilibrium and we can't get any higher. So co-current flow can get us to about 50% oxygenation level, which is pretty decent. But let's look back at our gills here. What actually happens is that the water is not flowing the same direction. What actually happens is the blood is flowing this direction, but the water is flowing this way. So it's actually counter current flow. So what we have is we have the deoxygenated blood. We're going to start out at 0, but the oxygenated water is coming in from the opposite end. So it's coming in starting out at 10, and we're just going to 9, 8, 7, 6, 5, 4, 3. I ran out of room. 2, 1, 0. We ran out of room, but we'll get there. All right, if I can. All right, so if I'm starting out at 0 right here, we've actually have the partial pressure pushing into the blood. So then we're going to gain 1. And now we're at 2. The partial pressure is still pushing into the blood, so we're going to gain another 1. Now we're at 3. We're still pushing into the blood, so we're going to gain 1. Now we're at 4. We're still going to gain 1, so I'm going to add another 4. Now we're at 5, pushing still into the blood. We gain 1. 6 pushes into the blood, so now we gain 1. 7 pushes into the blood, so now we gain 1. And then 8 pushes into the blood, so now we gain 1. And what do you know? We are getting almost up to 100%, 90, 100%, really high oxygenation levels just by changing the direction of the flow. So counterflow is much, much more efficient than co-current flow. There's also something called cross-current flow, which is not quite as efficient as countercurrent, but it can still be a positive impact because we will always have 100% oxygen level in the water, and then we will always have 0% in the blood. So we're always going to gain just a little bit of oxygen from this this cross flow current. But if I cross flow current several times, gaining just a little bit each time I cross the current, I can actually get up pretty high, maybe about 75% oxygen level. Still not going to be 100 like the counter current flow. All right, so let's review. Some organisms do what's called cutaneous respiration, where the gas diffuses directly across its body surface. This means that they're going to try to increase the body surface area as well as decrease the distance that these gases have to diffuse by either being hollow or having capillary beds right next to the skin. And many of them are going to use a digestive system or gastrovascular system to move this oxygen around their body. Then we have insects. Insects do two things. One of them is tracheal breathing with the spiracles. The spiracle is just an opening in the exoskeleton. It's connected to a tracheal tube system. Trachea branches off into tracheoles, which will branch into these little air sacs, which connect to the animal's body cells, allowing them to be oxygenated. We also have something called book page lungs. This is found in the arachnid family, so spiders, tarantulas, ticks, and scorpions. These look very, very similar to a book, which is why they're called book page lungs, because it looks like a bound book with the different pages, but they also look like gills, and they do behave a little bit like gills. You have the opening in the exoskeleton, and the exoskeleton is hydrophobic and has all these little hairs on it. So if these guys are dunked under the water, the first thing that they're going to do is just, due to the characteristics of their exoskeleton, are going to hold on to an air bubble on the surface of their exoskeleton. Also, all this has air held on into it because of this increased surface area here. So they can actually live underwater for quite a while. Scorpions is the most notorious for surviving underwater for about 10 days. So you do have to keep an eye out for these guys because they are highly camouflaged in the wild. And so if you're ever out hiking at night, bring a UV flashlight because their exoskeleton has chitin in it, which is UV reactive. They actually glow under UV light, which makes them much easier to spot when you're on a nighttime hike than this guy. All right, gills. Gills are great because we're going to be utilizing that counter current flow. Also, we have buoyancy, which means we can have tons and tons and tons of surface area on these gills and not worry about them collapsing. The buoyancy of the water will keep them lifted, and water will be able to readily flow through these gills. The water is going to flow in the opposite direction as the blood. And lots of different organisms have gills. Sharks, rays, bony fish. You have crustaceans like the lobster and crayfish. We have clams or cephalopods. These organisms all are using gills to breathe. Lungs are very widely diverse. For instance, we have these very rudimentary lungs by the salamander. You can see some initial efforts to increase the surface area with these folds, but just overall not very efficient. The frog lungs are a little bit more efficient. They have more dimpling and more folding to increase the surface area. Problem seems to be pretty resolved with the lizard. They have lots and lots of little bubbles, but still nothing like the mammalian lungs. The mammalian lungs have that alveoli sac, which really increases the surface area. So while the salamander lung is mostly hollow, these are actually pretty dense, and the only hollow part is the alveoli. Now, I do want a special mention about bird lungs. Bird lungs are super duper interesting. Here we have the trachea and the two bronchi that go into the parabronchi. The parabronchi is where all of the capillary beds are, but then we also have air sacs, and all these birds have different sizes of air sacs, but essentially what's going to happen is the air sac is going to get filled and then push air over the parabronchi only in one direction, whereas mammals, we breathe in and then we breathe out in two different directions called tidal breathing. So let's talk about these birds. It takes a bird 4 breathing cycles to completely exhale their breath. What that means is when you observe a bird, you can observe a bird breathing in and out rather quickly, actually. And so you might wonder why I say that they only breathe in one direction. Well, it's kind of like a bagpipe. He's breathing in and out, and he's pushing air into this sac, but that's not actually what makes the sound coming out of the horns. Just like the parabronchi, these horns, the air is only moving in one direction. So the air is only moving in one direction out of these horns, but he has this air sac, and whenever he wants to move air through those horns, he just has to squeeze the air sacs, and it squeezes air in that one direction across the horns. Meanwhile, he's breathing in and out all the time to fill the air sac. So that's the same thing that's happening with the birds. They're going to breathe in to fill up those air sacs, and then the air sacs will push air across the parabronchi in one direction. So the initial breath, breathe in, inflates the posterior air sacs. Then they're going to exhale. And when they exhale, the posterior air sac is pushed across, or the air in the posterior air sac is pushed across the parabronchi. And then they will inhale again, and it will fill up the cervical sacs, the frontward sacs. And then they will breathe out again, which the air will be exiting out of their trachea. So it takes 4 full breaths for this to happen, but when you look at it, it looks like the air sacs are inflating at the same time and the air sacs are deflating at the same time. All the time, there's only air moving in one direction across the parabronchi. So we can see this here in this GIF. You can see the chicken breathing in and out. And at the same time, these air sacs are filling and emptying with air, but they're filling with different air. So the new air is going straight to the back and the spent air is going to the front. All right, now let's talk about mammalian lungs. When we talk about mammalian lungs, we're primarily going to be talking about humans because it is the most visually relatable to us. You do need to know all of these terms for the exam, so do take a good look at this diagram to make sure that we know all of the parts of the human respiratory system. Paying special attention to the alveoli. The alveoli are the air sacs. Remember, they have to have a fluid over the top of them to help dissolve those gases, but as well as maintain structure. We don't want our lungs to collapse. Along the outside of the alveoli is the capillary bed, and the capillary bed is what's going to allow for gas exchange. But let's talk about the overall mechanism of the lungs. Each of the lungs is surrounded by a pleural sac. This is a thin membrane covered in fluid, and that fluid acts as a surface tension agent and allows the lungs to stay inflated. That pleural sac is also connected to the diaphragm and the intercostals. When the diaphragm moves down and the intercostals move out, the diaphragm is moving down and the intercostals are moving out, it causes the lungs to expand just by negative pressure. So we're inflating the lungs by expanding these muscles. And then when the diaphragm relaxes and the intercostals, which are your rib muscles, relax, it shrinks your lungs, causing you to exhale. So this is the method that you're going to be inhaling and exhaling, mainly with the diaphragm muscle right here, but also with those intercostals. And this wouldn't be possible without that pleural sac. Here is an example of that tidal breathing. You breathe in, the air in your lungs or the blood in your lungs is oxygenated, and then you breathe out in a tidal system. Here you can see the alveoli and you can see the oxygen being diffused into the capillaries. All right. Okay, so we are going to label the respiratory system. When we label the respiratory system, we need to find these different items. So bronchi. Well, I know that there are 2 bronchi. They're the main branches of this respiratory system. Trachea is the cartilaginous tube that we use for breathing. Diaphragm is the muscle that we use for breathing, and the larynx is going to be right there. All right, next, bronchioles. Well, if these are the bronchi, the bronchioles are all of these smaller branches throughout the lungs. And then it's asking us about the left lung. Now, this is my left side, but that's not the left side of the patient. The patient's left side is over here because remember, you're facing the patient. So that means this is the right side, and the left lung is going to be this one. Next is the alveoli. Here we see the alveoli sacs, and then last is the pharynx, which is also going to be in the back of this throat here. All right, if you want to measure lung health, we need to measure the tidal volume and the vital volume. So if you and I are just sitting here speaking, you're breathing in and out normally. This is your tidal volume, your normal inspiration and expiration. But If I were to vigorously exercise or breathe in as much as I could and then breathe out as much as I could, I would get this range. This is called the vital capacity. This is the maximum and minimum amount that you can breathe. Notice that there's always a residual volume there. We don't ever want to remove our residual volume because then those alveoli would collapse and pinch closed and we would not be able to breathe. So we need to leave a little bit of air in the lungs just to keep those alveoli inflated. In lab, we're going to be using a spirometer that looks a little bit different, but this is going to be a spirometer that will help to measure lung health. And this is your PSA. No smoking, please. Smoking is very dangerous. It is proven over and over and over again to lead to things like COPD, emphysema, and even lung cancer. And by the time the lungs get to this point, or even this point, they're no longer going to be efficient at oxygenating your blood. This is what a healthy lung looks like, and that's what we want to keep it at. So if you don't smoke, don't start. If you do smoke, try to cut back. I don't really have any personal advice for you. I'm just a voice inside a computer. All right, next let's talk about the circulatory system. So things like the sea sponge, the hydra, and the nematode, they don't have a dedicated circulatory system. Instead, they have something called a gastrovascular cavity, which they use for digestion. But they also use this liquid to oxygenate their body, which means their bodies generally have to be very thin or porous or just have an open cavity on the inside, hollow, like the nematode. Other organisms, as you get larger, have either an open circulatory system or a closed circulatory system, and they are aptly named. An open circulatory system is a series of tubes that are open to the body cavity. There is going to be a pumping mechanism, like a tubular heart, some sort of pumping vessel along this blood vessel in the back of their body. But again, these vessels are just open to the body cavity. So the heart or the pumping vessel is pulling liquid from the body cavity into the tube, which will then be spit out at the other end, starting the circulation over again. So it does circulate. It's just going to be limited in the size. We can't get super big with this because there's no control over the system. And this fluid is called hemolymph. So it's not true blood. It's not hemoglobin. It just is hemolymph, this body fluid or tissue fluid that also has heme in it. Now, a closed circulatory system is exactly what it sounds like. We have a true heart with true blood vessels that are closed, leading us even to smaller and smaller branches of those blood vessels called capillaries. Our first example of a true closed circulatory system is the Annelida phylum, or the segmented worm. Here we have true blood vessels with true blood. Now, the circulatory systems of different types of animals follow different patterns, but there's one main organization, that we have to have a capillary bed in the lungs or gills, and we have to have a capillary bed in the body. So we're going to show the circulatory system of these animals in this circular pattern. Remember, it doesn't exactly look like this, but these capillaries represent either the lungs or the gills, and these capillaries are going to represent the body where the fish needs to be oxygenated or the animal needs to be oxygenated. Now, there are lots and lots of evolutionary steps that help advance the circulatory system of these animals. We're going to start out by talking about fish, and then we will go through some different examples of these different circulatory systems until we land on humans. Some circulatory systems have a single loop, some have a double loop. And then we'll see that we're going to go from a 2-chambered heart all the way to a true 4-chambered heart. So lots and lots of evolutionary changes as these organisms get more and more complicated. First, we're going to go over the fish. Fish have one of the more simple organizations. They have a 2-chambered heart. So here you can see that thick wall of the ventricle and this like rudimentary atrium here. The capillary bed for their body is going to oxygenate their body, and then that blood is going to be pumped back into their 2-chambered heart, directly across the gills, and then back through the body again. Then we're talking about amphibians, and amphibians have a true three-chambered heart, but there's no septum in the ventricle here. So there's only one single ventricle, but there are two separate atria, which means that the blood is going to be mixed together. We have deoxygenated blood returning to the heart, and then it is pumped into the shared ventricle. Then it is pumped into the lungs to be completely oxygenated, but again, that is dumped into the shared ventricle. So the blood that is pumping through the body of the amphibian is half and half. It's mixed, deoxygenated and oxygenated. As we move forward into talking about turtles and some reptiles, we still have a three-chambered heart, but it is a septated three-chambered heart. It has half of a septum on the ventricle, which means that when the blood mixes, it doesn't mix as much. So now we have mostly deoxygenated blood going into the lungs, completely oxygenated blood coming back, mostly oxygenated blood being cycled through the body, so a little bit more efficient. Next, we have mammals, birds, and crocodilians have a true four-chambered heart with two atria and two ventricles. But first, what I want to show you, before we go into the mammalian heart, I want to show you the crocodilian heart. So the crocodilian heart has four chambers, but they have this area right in the middle. It's almost like a fifth chamber here, where some blood is mixed. So truly deoxygenated blood is sent to the lungs, and truly oxygenated blood is sent back to the heart. But then somewhere in here, there's a little bit of mixing. So the blood that circulates through the body is semi-oxygenated or half and half mixed. That does not happen in mammals. There is a true septate all the way through the mammal heart, leading to true four chambers. Let's look at the heart here. This is an example of the mammalian heart. We need to know all of the anatomical features as well as the path that blood takes through the heart. So let's see that heart beating. When the heart beats, the atria are going to fill up first, and then that blood gets dumped into the ventricle and pumped out. So let's start with the deoxygenated blood. Deoxygenated blood comes in through the vena cava, either the top or the bottom. Once it goes into the vena cava, it gets dumped into the right atria. Then it will be pumped into the right ventricle. Ventricle is easy to remember because it's shaped like a V. Once the ventricle pumps, the ventricle pump is a very strong, forceful pump. This blood moves past the pulmonary valve into the lungs via the pulmonary artery. Remember, arteries go away from the body and veins go towards the body. This blood is now oxygenated blood in the lungs and comes back into the left atria via the pulmonary veins. There's pulmonary veins over here and there's pulmonary veins over here, but they all four dump into the atria. Then it gets pumped into the ventricle. Now, this is the very strongest pump. It has the thickest muscle, and it is going to pump with great force through the aortic valve out into the aorta. This is the largest blood vessel in the body, and this is what is going to supply your entire body, your head, your arms, your trunk, your legs, even down here. This is the aorta. It twists over the back. All of your new oxygenated blood is going to be pumped through your body through the aorta. Here is a diagram with the labels. So if you want to review, you can look at this image. Let's look. All right, let's look at this image here. We are looking for the superior vena cava. Vena means vein, so I'm looking for something going towards the heart. There is a large vessel going towards the heart here and a large vessel going towards the heart here. Both of them are going to dump into this atria. So I'm going to pick the top one, superior. So this is the superior vena cava. Next is the pulmonary vein. I know pulmonary means lungs, so I'm looking for something to do with the lungs, and I'm looking for vein, which means going towards the heart. So this is a pulmonary vein, this is a pulmonary vein, this is a pulmonary vein, and this is a pulmonary vein, and all four of them are going to dump right here. Next is the left atria. So if I'm looking, this is my left side, but that's not the patient's left side. The patient's left side is this way, and the patient's right side is this way. So if I'm looking for the left atria, that's the left atria right there on the left side. And then we have the aorta, which is the largest blood vessel in your body, and it runs right like that and even down the back. So this is your aorta. All right, let's look at this one now. We're looking for the inferior vena cava. We said that this was the superior last time, so this is the inferior vena cava. This is asking for pulmonary artery. Arteries go away from the heart, and I'm looking for something to do with the lungs. So I'm going to go with this tube right here. It branches off into two different directions, and this is your pulmonary artery. Now we're looking for the right ventricle. Ventricle is shaped like a V, but again, this is the left side and this is the right side. So if I'm looking for the right ventricle, I'm looking for this ventricle right here. And then we have the pulmonary valve. If this is the pulmonary artery, the pulmonary valve is the valve that enters the pulmonary artery. Now, how does the heart actually beat? Well, the heart contains some excitable fibers. They are muscle fibers, but they have this excitable nature. They're called pacemaker cells, and they send an excitatory signal very reliably, starting out with the SA node or the sinoatrial node. When the sinoatrial node fires, it causes the whole top half of the heart to constrict. This is the P wave. Then it sends a signal to the atrioventricular node, or this second node right here. It will propagate a signal down into these Purkinje fibers, and these are also little nerve fibers or similar to nerve fibers that will then excite this large muscle at the bottom. Notice how thick that muscle is right there. When it takes a little bit of a delay, but when that signal reaches the Purkinje fibers, it causes a huge contraction known as the QRS wave, this huge wave right here, because the whole bottom half of the heart is constricting. And then we have the T wave, which is your reset, like relaxation. Everything's relaxing back to its normal shape. So to review, that is the P wave is when the SA node fires off and the top of the heart contracts. Then the atrioventricular node is triggered. There's a little bit of a delay, and then the Purkinje fibers are excited, which leads to the huge pumping of the QRS wave. So the QRS wave is this part, and then it resets at the T wave. So let's look. The cardiac cycle is going to happen with that lub-dub, the top pumping, then the bottom pumping. So what is the blood pressure? Well, blood pressure is the amount of pressure that these blood vessels are exerting, or that the blood is exerting on your blood vessels. So that's obviously going to be stronger when the bottom of your heart is contracting versus when your heart is relaxed. So the top number, often known as systolic, means the pressure that your blood is exerting on your blood vessels when your heart is pumping. And then the diastolic is the pressure exerted on your blood vessels when the heart is relaxed. So what is the top number? It's the systolic number, and it is when your heart is actively pumping. The diastolic is still pressure exerted on your blood vessels when your heart is more relaxed. All right, let's talk about blood vessels. We already mentioned that arteries go away from the heart and veins go back towards the heart. But why are they different? Well, arteries have a very thick connective tissue and muscle wall around the outside of the artery. And this helps it to resist the intense amount of pressure that the heart places on these blood vessels. So they're reinforced. By the time that blood moves through your capillary, it's no longer moving with as much force. So now we have to figure out, how do we get it back to the heart? So while the artery is moving away from the heart, the veins have to move back towards the heart. And since they don't have a heart, basically a lot of them are just going to rely on your body movement, like the muscles in your legs. When they contract and move, it actually pushes the blood up through these blood vessels, and then they have one-way valves. So that's another major difference. Arteries do not have valves, whereas veins have these valves to prevent backflow. Sometimes or occasionally backflow does happen, and it can change the shape of your veins, leading to this varicose vein, because there is some backflow and those valves are no longer working properly. This can lead to some cardiovascular diseases. Buildup of plaque and other fatty cholesterols can cause a buildup inside of your blood vessels. As the buildup gets bigger, it reduces the blood flow and can even catch platelets, which then can cause a blood clot or constriction of blood from necessary areas. So atherosclerosis and arteriosclerosis is going to be hardening of those arteries there. Now, let's talk a little bit about blood before we go. Blood is considered a connective tissue, and what we focused on mostly is the red blood cells. But there are also white blood cells, plasma, and platelets. Blood functions for lots of different purposes, and it is made in the bone, which makes it make a little bit more sense why it's considered a connective tissue. Blood functions to transport gases and nutrients and waste, but also to regulate your body by facilitating heat exchange, maintaining pH balance, and distributing hormones throughout your body. And it also helps with protection, because the blood cells, or the white blood cells, are a major part of your immune system. And also platelets help to protect you from scrapes and cuts. All right, so how is blood formed? The short answer is that it's formed inside the bone marrow of your long bones. This is a picture of your femur. That's your largest bone, but you're producing blood all over your body at all points in time, because your erythrocytes only live for about a month, month and a half. After that, they're too far damaged and they need to be replaced. So your bones are constantly making new blood cells, about 2 million per second. But that's not the only thing it's making. It's not just making erythrocytes out of these blood stem cells that are located inside of the bone marrow. We're also making white blood cells and platelets. In fact, if I took your blood and spun it through a centrifuge, it would only be about 20% red blood cells. It would have a small layer of white blood cells and platelets, but it would be mostly plasma, which is essentially water with some dissolved gases, nutrients, and ions like copper, calcium, and sodium. We also have some proteins in the blood plasma, such as albumin and some globulins, and we call that the serum, but it's all part of the blood plasma. Platelets, of course, are important for clotting. They produce a fibrin protein, which can gather together and clump together and create a barrier in the case of damage. White blood cells are also called leukocytes. This is a very, very small proportion of your total blood, but they do become very important for immune response. So we have two main types, agranular and granular. Agranular includes monocytes and lymphocytes. And then granular leukocytes include neutrophils, eosinophils, and basophils. These are used for purposes. For instance, these are used in an inflammatory reaction, where this is used more in an immune, secondary immune reaction. Now, back to this slide. We've already seen this slide, but I just want to remind you that these red blood cells called erythrocytes contain hemoglobin, and hemoglobin has iron, which gives it that red color. But hemoglobin is not the only pigment that helps carry oxygen. So there are other pigments, such as red, based on iron, from hemoglobin. Here you can see the iron right there in the center. But we also have some copper-based heme called hemocyanin. Here you can see the copper right there located in 2 locations. Chlorochronin, which has a light green color, when it becomes oxygenated is more of a pink color, is also based on iron and is very, very similar to hemoglobin, just slightly structurally different, such as this oxygen right here. Last is also iron-based is the hemorythrin. It has iron right here and here. It's more closely structurally related to the hemocyanin, but instead of copper has iron, and it has a bright violet coloration. Is found in some marine worms. Now, one famous example of blue blood is the blood of the horseshoe crab or the Chelicerata. Okay, so this is an example of evolutionary stasis. These horseshoe crabs, they're actually closely related to arachnids, and they have a hemocyanin, a copper-based blue coloration of their blood. And this blood is highly valued in the medical community. These horseshoe crabs, they gather in very large numbers on the beach when it is mating season. So all of these guys are getting together ready to mate, and then we scoop them up and bleed them for their blue blood. And this blue blood is very valuable, especially with endotoxin detection. So it can be mixed with different types of medicines or to detect different types of toxins before they give out a medication. All right, that's it for today. Don't forget you have the optional chapter homework that you can turn in for extra credit. If you need the answer key to that, you can contact your SI leader. Also, don't forget to do the chapter laboratory write-up. The video is posted online, and you can click this link to get the lab write-up. Also, don't forget your chapter quiz. This links back to Blackboard, so you don't have to click this link, but make sure that you take your quiz by 5 p.m. on Friday. Have a great week.